My name is Julia Schaletzky. I'm the Executive Director for the Center of Emerging and Neglected Diseases. I'd like to welcome you all today for this coronavirus hackathon. I'm so glad to see so many people from all over the world join today virtually. We have totally pivoted from an in-person event to a virtual event, and Isabelle, you already met her, really did the majority of the work on this. I'm very grateful. Thank you, Isabelle. Giving you a good clap here. You made this happen. And it's very exciting to be here. So I'll give you an introduction first about what CEND is, what we do, why we came up with this idea, and then I'll hand it over to Isabelle to give us an overview of the day and how this is all going to work logistically to make sure everybody gets talking and brainstorming with the right people, and we can come up with some ideas around the questions we've asked. All right. Welcome everyone. I'm first going to give you some overview what CEND is. We have a team. No, it's on the automated. No, stop. I think it just. So we are a team of scientists and project managers working on combating emerging and neglected diseases. Our faculty director is Jeff Cox, and Laurent is our associate faculty director. Jeff Owen used to be the Dean of Biological Sciences here. Celine is our grant writer. Eddie is our screening scientist. Isabelle, you already know from this project. She's our project manager also for the Alliance of Global Health and Sciences, and Sarah is our scientific advisor. On this particular project, the majority of the work has been done by Isabelle together with Shreya and Louis, two students that have helped us out very graciously on setting this up, which I'm very grateful for. And they really made this happen. So our mission is to connect researchers interdisciplinarily from diverse disciplines, geographic regions, and sectors to foster innovative, interdisciplinary, and collaborative research. I think I'm just going to log out here because it is on some timer. And okay. And we're trying to catalyze research with the center. We have a drug discovery center to do a translational science to connect the basic research that's going on campus with more translational efforts. Also, like what we're doing today in the hackathon, really interested in making happen. So small molecule discovery center is just one part of that. We also do have yearly symposia and events. We teach classes, so we're trying to enhance the profile of emerging and neglected diseases in public. Right now, I would say the profile is very high, even without our work. We've been, pushing pandemic preparedness and things like that for years, but I would say in general, few people listen, but now everybody is really interested in it because of the active COVID pandemic that we're having right now. What do we offer in general? I don't know, many of you probably are, not everybody's associated with UC Berkeley, but within UC we have graduate student fellowships we give out, and also internship opportunities that are open to everyone who's qualified. We do also administrative and project management within the center. We have a grant writer, and we can help with like training grants and projects that need a little bit of administration that's often not supported in academic labs. So we're helping to do events, conferences, hackathons, provide international training experience and research. We have strong ties to Africa and have active collaborations there. We have a lot of experience with training grants, writing and administering them, and are particularly active for supporting underrepresented minorities in STEM. So that's some work we do there to support academic research and bring some more opportunities to campus. For translational science, which is more our focus today, is we have the state-of-the-art high-throughput drug discovery center, and we provide also hands-on biotech training. We have some help that we offer with startup formation. We offer sample management capability for biological and chemical samples, and have a large interdisciplinary network of investigators that we can bring to bear, and we're trying to make use of that right now for the COVID epidemic. So what have we been doing on COVID as a center? So CEND was active early on. The first time I heard of this outbreak was during our symposium, actually on January 11th this year, when all the epidemiologists we had invited and people who were very growing outbreak in Africa, all were talking about this coronavirus, and I hadn't really heard much about it at the time, but I was remembering that how it was very concerning to see all these very experienced outbreak and pandemics, people being really worried about it. So I think that rang true definitely later. At the time, I wasn't really sure what to expect from this. But CEND has been involved in a variety of ways to generate a response from campus on COVID. So we're working actively right now on helping to build clinical diagnostic capacity on campus. We are working on fostering interdisciplinary research because a lot of the questions we need to solve require researchers from different areas to work together. We also managing an established network of virologists called Bay Viro. You can find it on our website here. We connect virology research throughout the Bay Area, up to UC Santa Cruz, and it's a great resource now because we kind of kept that going. We did yearly conferences and make sure people work together, but this is a great tool now because everybody's connected, and so we can also coordinate who's doing what on the COVID response. And to that end, we've put together a list of resources like cell lines and plasmids, other tools and funding opportunities currently open for research around COVID on our webpage, and that's available to the community. If any of you have things to submit there, resources you have and you can offer within the Bay Viro network or the broader community, feel free to submit something. There is a button there to do that. So that's how we're getting involved. I'm also personally doing a lot of outreach, press work, everybody in keeping the public informed. We've hosted several events with the Department of Public Health with Q&A around coronavirus so that people can have a voice and also hear high quality, scientifically sound data. And all this is right now a very busy time for all of us. All right, so in general, we're interested if anyone has beyond, in the future, who would like to have some help administering or putting together a grant with an administrative component. We're really interested in doing that if it fits where we're going, center and program grants or larger interdisciplinary projects. We can help, help to get it going. And then also anything that involves drug discovery approaches, academic science that's more translational or leads to startup formation, we can help with. And then we also publicize anything that is related, from CEND faculty and associated groups. Also, all of you, if anything comes out of the work that you do today, be happy to feature it and put it on the website, and make sure you get more publicity around the new findings. So we rely on people also to reach out to us to let us know if there's opportunities that could fit with our mission, or people would like to volunteer for us. I encourage you all to sign up for our newsletter. We don't send a lot of emails. It's basically just invitations to the yearly symposia that we do. We try our best to keep them free, and they're always very interesting, focused on emerging and neglected diseases and on virology. All right. If we ever get out of the lockdown, also feel free to come by and visit us. We're on the University of California, Berkeley campus in the Li Ka-shing building. All right. That's it. Here's our Twitter handle and ways to keep connected online. And with that, I'll hand it off to Isabelle to speak about today and what we can expect in terms of agenda and outline.
Great. Thanks, Julia. That was good. Okay, so I'm going to go ahead and go into what to expect over the next 2 days, what today's going to look like, and what our expectations are going to be as far as how you're going to be working in your teams, what the end product is. So let me go ahead and share my screen. Share. Okay. All right. Can you all see the PowerPoint?
Yep. Yes.
Cool. Okay. Okay. So here is the 2020 COVID-19 hackathon. hosted by CEND. So I'm assuming a lot of you have participated in hackathons before, but for those of you who have not, I'm going to talk to you a little bit about what a hackathon is and what this looks like. So it is not a traditional hackathon like you might think of, where it's a bunch of computer scientists sitting in the same room for 48 hours, drowning in coffee, trying to hack into some sort of system. The idea of a hackathon is to take a complex problem with people from a lot of different backgrounds and figure out how to hack that problem and implement solutions that may not have come out of, you know, a meeting with people who only come from the same backgrounds or who aren't having that collaboration. So, you know, bringing people with different experiences, different insights, and different expertise to talk about these issues, then in this case would be the COVID-19 pandemic. So this year we started out working on doing diagnostics for the hackathon, because at the time when we were planning the hackathon, that felt like the most relevant issue. This pandemic has obviously been evolving very quickly, and we found that we should open it up a little bit more to address other issues that have arisen as a byproduct of the pandemic, and that will still include diagnostics, but we're also looking at medical device development, comorbidities, and using data to strengthen health systems. So you have been broken up into groups based off of what you indicated your interest was when you confirmed your participation in this hackathon. If you did not indicate, if you said that you had no preference, we likely put you on a team that was kind of needing someone of your background, and I will talk about this a little bit later, but that is not, these teams aren't necessarily set in stone. You will have opportunities to talk to other teams if you want to, or change teams. But so let me talk about the schedule for today. As we have already done is the first part, which is opening remarks and introduction to CEND by Julia. And then I am now going to the outline of the day, the expectations and questions. Depending on how many questions everyone has and how quickly we get through this, we will go a little bit over the global state of COVID-19. I'm sure most of us have been keeping up with the news pretty frequently. So, but for those of you who maybe have not or would like a little bit more of an overview, that will be a chance for us to go into that a little more. And then at 3 p.m., so at this point, participation in the Zoom conference is optional. You can go off into your groups if you'd like, but we are breaking into 2 groups based off of the diagnostic development group and the respirator/medical device group. Those of you who are working in data or in health systems, or maybe aren't as interested in participating in the panel, do not have to participate. But it is a good opportunity for you to talk to people who are actively working in the fields of diagnostics or medical device design and development. So my student assistants will be sorting you into 2 rooms based off of the teams you've been assigned to. If you want to be put into a different group for the panel, we can do that. Just go ahead and shoot us a text after you've been put, or an email or a message on Zoom, and let us know you want to go into the other talk. So the diagnostic talk will be hosted by Celine Perrier. She is the grant writer for CEND, and that will feature Lisa Diamond, Tara DeBoer, and Heather Koshinsky. And then the medical device development will be moderated by Julia Schaletzky with Dr. Gerlin Vernig and Lawrence Lynn and Robert Miras. Those talks we expect to last about an hour, including a Q&A session. And then at that point, you will be broken off into teams and you can go off to do your work on whatever platform you've chosen. So at 8 p.m., I am requesting that all groups submit their initial project proposal. So this is likely going to change over the course of, today or tomorrow, but we at least want to get an idea of what you've decided that you want to be working on in relationship to the topic that you were assigned. And this will be posted on a Google Doc with a primary team contact and shared with the entirety of the conference participants. And the reason that we're doing that is there's some people who might be on teams where they realize maybe they aren't as interested in the project that people have decided that they're working on, or they realize maybe their background doesn't align with what that project needs. And this is an opportunity for people to connect with other groups and say, hey, this is my background. Do you need someone? Oh, someone's joining, admit. If there's someone that you need from my background, I'm available to work with you. And it's also an opportunity, maybe you like the team you're on, but you also just want to see what other people are doing, and maybe you can have some insights or give them some guidance. And if you want to do that, you can as well. So I will be sending you a Google Doc with this PowerPoint after this. And that Google Doc will be where you fill out later tonight your initial project proposal, as well as your designated team contact information. Okay. Oh, why is there we go. So tomorrow is going to be mostly just you guys all working together. We will do a check-in in the morning at 9 a.m. where I will be available to answer any questions that you maybe have that either if you shot me an email and I haven't answered it, or you just are stuck on something, there's an opportunity for us to check in. And then that should last about 10-15 minutes. And then for the rest of the day, you all are free to break out and work together. Our grant writer, Celine, will be hosting a panel at 12 p.m. tomorrow on accessing COVID-19 funding opportunities, which I'm sure many of you might be interested in. She'll be talking about what the current opportunities are and how the best way to navigate applying for those will be. Someone else just joined, admit. And that should last about an hour. And then we expect that your final projects and proposals will be submitted by 2 p.m. to our judging committee, who will be working to review them, have them reviewed by 5:30 p.m. So that's the schedule of the next 2 days. Please let me know if you have any questions about that at the end. So a little bit more about logistics. What platforms will we be using to participate? So the videos and the conferencing will all be done over Zoom. You've all gotten a Zoom link by now. If you're having difficulties accessing Zoom, or you have someone who you know who was supposed to participate, but for whatever reason they can't access the link, please let me know. And then how you'll be working with your groups, I am going to leave that up to you. Everyone has a preferred platform. Some people prefer talking on the phone. Some people want a video chat. Some people want to talk via Slack, and some people prefer email. So that will be left up to the teams. I discussed how teams have been formed already, and I have discussed how to change teams. I will note something that myself and the people that we're working on creating teams with did, is there are some people who came into this conference who indicated when they applied for the conference they had an idea that they already wanted to work on. So those people we assigned some other people on their teams to as well. So if you know that you indicated that you had a project coming in with, us assigning someone to your team doesn't mean that we were swiping your idea away. It means that we had people who were willing to work on any project who had a background or an interest that we thought would align with yours. And then for the other teams, we assigned you someone who has some sort of leadership experience in the area that you are working on as your team lead. If you have not heard from whoever your team lead was or from your team at all, please let me know, and we can assign you another team, or I can reach out to whoever it was that was supposed to be leading your team and figure out what's going on. And yes, as I mentioned, you can change teams.
Hey, Isabelle?
Yes?
How many teams are there?
There are 13 at the moment.
Okay.
And there might be more, or there might be less if any teams decide to combine or split up. We would try to keep them minimal so the judges don't have a lot of projects to review. Yeah. And then the final product of the hackathon is a project. So let me go into the project and proposal submission a little bit. We've got a question. Chat. How many teams on medical devices and other categories? So, was that what you were asking, Matt? So there's 13 teams in total. They might break up into more. I think we've got 5 working on diagnostics, 3 working on medical devices, and 5 working on data. 6 working on data. I'll have to double check that, but that information will be shared later on as well. Once people have confirmed what their team projects, the rough draft will be. Okay, so for your project / proposal submission, I think that every project is going to be unique, and in that sense, I don't want to restrain you to how you choose to present your final proposal. Some of you are working with data and you might not, and so, asking for you to present photos of a prototype or a paper might not work as well as having you do some sort of visualization. So the media by which you choose to convey your idea is up to you and your team, and how you hi Carter, how you think that it will best convey the idea that you're trying to tell. So that's, again, up to you. Be creative. This is, because it's so open, this is an opportunity for you to branch out a little bit. Please try to keep it no more than 10 minutes to review. People will be, let's see, we've got a question. So, okay, I see your questions. I'm going to answer them all at the end, just to let everyone know. So I'm not ignoring you. Okay. So no more than 10 minutes to review, just because we do have panels of judges and there are going to be over 10 teams, which means that they need to thoroughly review all of your applications. So please keep that in mind, that you want whatever you're proposing to be as precise as possible. We also want to make sure that you include a rollout or implementation strategy for whatever your findings or your project is. So if you are working with data, how is this data going to be relevant to the response? What are you going to do with it? And how are you going to make sure or try to get that data or those findings implemented in policy or project? If you are doing a medical device, how do you plan on getting that through the FDA? What are the steps you're going to take? So these are all things that are going to be taken into consideration as well when we're judging. So please keep that in mind, that beyond just submitting your project proposal, you also want to think about funding. You want to think about any approval processes that you have to go to for getting a product on the market, etc. Okay, and then how will my project be judged? So first of all, we're going to look at need. There's obviously a lot of needs in this pandemic, and there's a lot of things that need to be addressed. Have you articulated how this project will fit a specific need within the pandemic response? Scientific feasibility. Is this project grounded in data and science? Have you thought about how realistic this is to implement? Have you looked at the papers? Have you read about the research that's already being done in this area? As you may have saw, my Shreya and Luis did a good job of getting some resources and data sets uploaded onto the box, which was shared with you in the email I sent out last night. So hopefully you've been able to look through that. That is definitely not comprehensive, but we thought that would be a good starting point for you. Originality. Is this idea novel, or is it a duplication of efforts? So if you're doing a diagnostic product, is this something that's already being done? And if it is something that's already being done, how are you improving upon it? Because you can take an idea that's already being tested and make it better. But what we don't want to see is for you to be taking Gilead's therapeutics for remdesivir and just copying that idea and submitting it. So the idea is to create something that's innovative, new, and sometimes it is a good idea to take from what's already being done and improve on that process. But we will be looking at originality when we're judging these. Execution. So have you considered how this idea or your findings will be implemented or integrated into current solutions, which I talked about a rollout plan? And then sustainability. Is this a short-term or long-term solution to the pandemic? So how will this last beyond the implementation phase? Is it something that we can keep up and rolling if we expect there to be another outbreak, etc.? Are vaccines and therapeutics excluded categories? They're not excluded. We did not include that in the call out just because at the time we were focusing on diagnostics. If you are interested in working on therapeutics, you are totally, this is open for you. We had focus areas and we had people that we brought on to be mentors in those areas, but that does not mean that you're excluded from working on something else that you feel like your background is more fit for, or that you feel like there's a big need for. Yes, when you're working on the data, please provide a GitHub link along with your project submission. Will there be pitching of ideas or evaluation only based on material? We will, unfortunately, because of time and internet restraints, we will only be pitching ideas based on material. So some people here might not have the best internet access, and we want to keep this equitable for everyone who's participating across the world. And so we didn't feel like that would be conducive, because some people in Berkeley might have better internet than some people who are participating elsewhere. And also for time, we only have the judges only have a certain amount of time that they can commit. Have all the teams been assigned a leader? Yes, you should have. Or if you were someone who submitted with a project outline when you submitted your confirmation, then that you and your team are technically leaders. If you haven't heard from your lead, please message me separately about that. So we are going to now welcome Julia back to give a short overview of the current COVID-19 pandemic. And from that point on, everyone will be broken off into their groups. So
Right.
Take it away.
Okay, so the current COVID pandemic, I think we all in the middle of it. It's been quite a ride, I think, for all of us, even people who've worked in epidemics fields for a while. Kind of shocking how fast it has been spreading. And maybe the thing we can look at it is first the current numbers and some interesting things we can deduce from that, and then we can delve more into epidemiological questions, as well as what the status is right now for diagnostic therapeutics and vaccines. Going to open up a site because I've been working around the clock on the COVID response, so I haven't really made slides for this, but I think we should just look at the Worldometer site because it is quite good. So I will share that. I think it's here. All right. Can you all see it? Yeah. Worldometer info, all the graphs. Yes. Matt, give me the thumbs up because you're right under me. Yes, excellent. Good. So this is a pretty good site if you want to check it out. The Johns Hopkins site is also very good. They tally all the cases that have been diagnosed properly worldwide. I think here's the first caveat, because we only know for sure the ones we've tested, and since testing is such a bottleneck in many countries, I think it's an underreporting of the actual number by a large amount. We currently have almost 500,000 cases diagnosed, 20,000 deaths, and then 113 have fully recovered. So, you know, we are in a pretty scary part of this curve. It is a full-blown exponential this growth of new coronavirus cases. If you look at different countries, what's striking, and you probably have seen this in the news, is that the mortality definitely is very different between countries. I mean, particularly Italy, you can see has more than 70,000 cases, more than 7,000 deaths. That's a mortality or a case fatality rate, as they call, of 10%, which is massive for this virus. It's way more than anyone thought. Similar in Spain, also very high. Germany, however, almost 40,000 cases, just 200 deaths, so very low. And this is interesting. People trying to figure out now why some countries have high case fatality ratios, others have low ones. It probably has to do to some extent with how overloaded the hospital system is, but probably doesn't explain the whole story. It also can have to do with facts like how much smoking is happening in a country and comorbidities like that. But I think I mean, I'm still not convinced by any of these things to really explain this whole massive, large difference, because Italy is also a rich country with great hospital infrastructure, and I don't really know what's going on there. People are trying to find out. Some of it is the age population. Italy has the oldest population on average within Europe. and we know that the virus hits older people harder, and then also smokers and people who have diabetes, things like that. But I would say let's start maybe with China. We had the first case in China. The first case was probably middle of November last year. That's what they say. And then in early January 2020, it was communicated. In China, we have kind of encouraging data at this point because we see that we have reached a plateau in total coronavirus cases. The daily new cases really went down. And I think the last couple of days, they only had 2 or 3 new cases. So that is really good news. They have closed down already all the COVID hospitals that were built because there's no more demand. So that's the good news of the day, and active cases going down. So China's quarantine and lockdown methods, and so far been quite successful in reaching a plateau in this curve. The other country that's really interesting, I think, is South Korea, because they ran everything very different from what we do here. They never did any shutdowns, lockdowns, but in fact, the first 3 days of the pandemic, they tested more than 130,000 people and were able to also achieve a plateau of total cases and really only very few daily new cases currently, without any large-scale quarantine in the country. So also very encouraging data. But they reacted very proactively, immediately assembled a task force the same day that the first case was known in China, and just already building testing capacity immediately, and testing more than 150,000 people, which is within the US now, I mean, far out of reach even months into this epidemic. So then the United States, where are we? For us, looks a little different. We're right in the scary part of the curve, so full exponential growth. And we have lockdowns now in many areas of the US, including California, that are very strict, but we don't see the effect yet because it takes some time for this to manifest itself, probably at least 2 weeks, which is normally the time that the virus takes to infect. And also incubation time is around 6 days. So in theory, if we do a total lockdown, nobody sees anyone, and you have only small family units, you could think if everybody is completely separate for 2 or 3 weeks, we should have eradicated this virus. But since this is not happening easily in a society, we still use this method to lower the load on hospitals and just slow the infection. So I hope that we will see soon a decline of daily new cases in the United States. We are still going up, and we also see the exponential curve of the daily cases, active cases, and also the deaths are catching up now. This is normally a little lagging behind the new cases because it takes at least a week for the disease to get bad enough that you need intensive care. But it's also pretty grim here. We have a real increase in the daily deaths now because it's kind of in the full-blown phase. So I think all of you have followed to some extent this kind of, I don't know how to say this in a politically correct way, but the failures of the government to deal with this decisively and in a fast fashion as this was emerging. I would say it was very interesting what happened on the diagnostic testing capacity. We have, I would say, enough capacity in the US in principle to test a large swath of the population, but we can't make it happen because mostly regulatory issues around clinical testing that are so restrictive that it's very, very, very difficult to build clinical research capacity because it's so heavily regulated. And we already in peacetime have operated at capacity, and now a big epidemic hits, they have to set up a new assay they haven't done before. Everything has to happen immediately, and then they have to do large scale testing. It just wasn't possible in this environment without unleashing, the academic sector, and they just haven't done that fully because it's still highly regulated, and we're just building, testing in Berkeley, and the compliance issue is a major issue. It takes forever. Nobody really knows what the rules are because they change daily, and everything is under emergency declaration, but gives you a lot of power, but you need someone brave enough to also say, this is an emergency situation. From now on, all you labs can do testing, and I take responsibility for it. I haven't seen that either. So everybody is still trying to cover their behind, and it's like we're trying to make sure that everything is super compliant. And that also means we have less testing capacity. And we even dealing with, maybe should do a hackathon on regulatory innovation, because it's also needed badly. I mean, every day I learn things where I'm just like, why is it like that? For example, if you do a research study testing people now in the United States for coronavirus for research, which is exempt from all these clinical trial regulations, you are not allowed to share the results with the people in the cohort. So if you test 3,000 people that are asymptomatic, you want to find out how many have it, and then you find 8% have it, legally you're not allowed to share that information with the patients, which to me doesn't make any sense. And but it is only a doctor ordered test performed by a doctor can be communicated by a doctor. So it's a federal law. And that really hurts us in this epidemic, because either you're compliant or you just have more growth of this virus. So it's a bad choice. So little side thing here about diagnostics and why it's so hard in the US, particularly to test it. South Korea, all this was no problem. They just did it. And I think that's what we should have done. The CDC had some time, because this virus emerged in Hubei province abroad. We had a couple of weeks where we didn't see much of coronavirus in the US, although we don't really know. Maybe it was there already and we just didn't know. The CDC developed the test. Instead of just applying, what other people had developed, they chose to develop totally their own test and different primers and build it all from scratch. And then unfortunately, part of it didn't work, and they had to recall a large amount of test kits. So that was one problem. The other problem was that they, in the beginning, didn't allow anyone else to do any testing. Everything had to go through the CDC, and that was not great for curbing the spread, because they were very slow. It meant for you personally, you had to take a sample of a patient in a doctor's office that had to be mailed to Atlanta, where the CDC is. And then the turnaround was, in the best case scenario, 5 days for you to get the result. So in these 5 days, you keep infecting everyone, right? So that's one reason why we want to talk about, real-time diagnostics, ideally, point of care diagnostics, which are also much easier to handle from a regulatory perspective, because they exempt from a lot of these rules regarding CLIA and things like that. It's more like a pregnancy test model where you can test at home and know what's going on. So that would be the ideal. And that's where this idea came from, because in order to curb an epidemic, you can do it two ways. Either in the beginning, when it starts spreading and it's not many people, you just test large groups of the population and you find the ones who are carriers and you quarantine them, which is the approach they took in South Korea. Or you just don't do anything like we did, and you limit testing capacity with a lot of rules, and few people get tested, and then you see the disease popping up everywhere before you actually know how it's moving, because you haven't tested people. And then the only option you have to curb the spread is to do draconian quarantine measures and put everyone on a lockdown. And that's what we're doing now at extreme cost to the economy and GDP, and the long-term effect of this remains to be seen. But this is what's going on in the US with the testing, why it's been so slow, mile high overview. And currently a lot of places are trying to ramp it up, and we got, government kind of with a lot of fanfare announcing everyone can be tested now. I'm not aware of any testing lab in the Bay Area where anyone can be tested. I mean, they still have a lot of rules. Only certain patient population qualifies really to be tested. And even then, turnaround now at Quest is around 7 days to get your result. So this is almost the whole case. Coronavirus maybe lasts for 2 weeks, and 1 week, first you have to get sick enough that they give you a test, and then you can wait up to 7 days to get the result, because they don't
Hey,
have
Julia.
capacity. Yes.
I have just a couple of points. I agree with many of the things that you're discussing. And I think one of the things that hasn't been highlighted is the difference between South Korea and the United States are the healthcare systems. You have a nationalized healthcare system versus ours, which is extremely segmented. And this comes down to the complications in training, what you were just talking about. No longer is it a government agency that's rolling out. Right. In the US, there is the hurdles of educating people and getting them up to snuff of how to run a test, a diagnostic test, is not trivial. It's one of the biggest barriers to a diagnostic company. And so I just want to highlight that there needed to be some kind of central entity that helps in that adoption implementation component. And I think that's one of the big things that's lacking in the US. And the regulatory standpoint is, as a diagnostic, as an individual who's going to be taking a lot of responsibility on creating a diagnostic, we think about the impact of false negatives and false positives on people. And the FDA's number one job is to assess the risk of a diagnostic and its impact to the user and to the patient. And so the danger in letting out too many tests, it can be really detrimental on a bigger scale. And I agree that there's a lot of things that are not running optimally, but I do want to, I actually have interacted with the FDA quite a bit, and there really are, I just don't think that they're big enough to take on the load of all the diversity of the healthcare system and the different types of tests coming in the door. They're completely overworked.
Yeah, I agree. I mean, and currently all the government agencies dealing with this are completely, I mean, you can barely get a response out of anyone. And that's right now at the time when we really need some clarity around these regulatory questions. Yeah. I mean, the false positive and false negative question is an important question also to think about for all of you who want to contribute to the hackathon for diagnostics today and tomorrow. It's true, we don't want to have false positives particularly. However, I think, I mean, I have to say that the way it was rolled out by the CDC was kind of absurd, because they were supposed to be the ones to do it right in order to avoid false positives. And then a third of the test kits they delivered were false positive. So
that was ridiculous. That was a complete fail.
Yeah,
I agree.
It kind of makes you wonder about these rules, because in principle it's true, but then, you know, if they can't get it right in the time of need, I mean, we can't really stop everyone else. I mean, I think personally the prime it was a lot about the primer design. I think that they chose, and you know, other countries have developed a bit better primers. All the professors that do qPCR that saw the primer set from the CDC, they were like, why would you do that? And so I don't know. I think that there was certain questions around it. They could have done that better, but it's critical, I think, for any diagnostics developer, because you don't want to develop something where the signal isn't clear, and you want to make sure you don't get false positives. I mean, false negatives is a little bit a different question, I feel, because it depends also on the sample collection and things like that. Even with your best effort, sometimes maybe you have a false negative. But the false positives you should avoid. And I think from a public health perspective, even if you have some false negatives, it's still better, as long as you find on the false positives, it's still better to test, even if you only capture, you know, 80% of the sick people, versus just not test and not capture anyone, which is what they've done here. So all good input, and I think this is something we have to keep in mind with diagnostics. Particularly, it's important to have something that allows you with good certainty, precision, accuracy to distinguish positive and negative. I mean, it's one of the key things. There's nothing more frustrating than if you might have a serious disease and you do some kind of strip test and you see there is a band and it's slightly gray, and you can't decide if it's there or if it's not not there. So you need to, that needs to be addressed for all kinds of point of care diagnostics that we consider. And there's ways to do this, as Tara is doing. and she will tell you more about later, right? So that's the part on the diagnostic. What else is going on? Currently, we in the middle of it. There is, it's kind of, you can be of two minds if how much of a difference diagnostics still make, because right now, if everybody gets it soon, maybe then what they did in China is they just stopped diagnosing people. They just treated everyone as well as they could. But in general, I think it is good to develop diagnostics also so that we prepared for the future, because I think really what we're hoping to do is maybe have systems in place that can be more easily adapted to another pathogen, because right now everything is driven by the PCR system, but we don't have much going on in terms of point of care that's out on the market. And if we can find something more universal there, would be fantastic. The other thing that's happening is vaccines. It's probably the most advanced field. We haven't included that here because anything we develop here probably won't move the needle much in terms of timing that it takes. We have already a vaccine in clinical trials right now. The Moderna vaccine is an mRNA vaccine, and there's other efforts also of recombinant vaccines going on. Moderna was interesting because they got away with not doing any animal studies for the vaccine, which to my knowledge has never happened before. So they were able to push that through, and they dosed, a week ago or something, the first patient in Seattle, and it was well tolerated so far. So with mRNA vaccines, there's different ways how you can make vaccines for coronavirus. The best vaccine would be a live attenuated vaccine, but we don't really know how to grow this virus at high titer. So it's not so easy to do that. And there's also safety concerns, and you need a biosafety level 3 to grow it up. And all this is a bit complicated. But normally you get excellent immunity from these live attenuated vaccines. The next thing that many people do is to do a recombinant vaccine. So there you try to pick out a surface protein from the virus that is conserved and hopefully won't drift too much with mutations. And then you take this, express it in a system, tissue culture. Eggs are used for flu or something like that, and you're trying to in bacteria, the eggs is for the live one, and you purify from yeast or bacteria basically the recombinant vaccine. You inject it in antibodies form. And then the third version, what Moderna is using, which is faster, is to purify mRNA and then have your body make the protein that becomes the antigen for antibodies to form. And that is faster because the way to manufacture a GMP batch of mRNA is easier than to make a protein GMP batch. So they were able to do this very fast and inject it. The problem with mRNA vaccine is more that sometimes it's not very immunogenic, so the antibody response is not very strong. So that remains to be seen with that one. I mean, right now it seems like it's safe, and I hope they generate more data around that, but the efficacy is another question. However, there's always debate about timeline, that people say 12 to 18 months for vaccine. That's true for in normal cases. Right now, it could be a lot faster as long as there's regulatory relief. We have already, they've did away with the mouse studies also because currently there is no mouse model for this disease. So the mice first have to be grown up from SARS because a while back we had the SARS epidemic, and it's also coronavirus. So a lot of the tools have been developed, but these mice weren't currently used. So we're all frozen down as embryos. So they first have to be implanted, then a breeding colony has to be grown up. All this takes time. It's not available right now. They're just taking pre-orders. And so for that reason, I think they were able to push it through to go right into humans. And with vaccines, though, the question is it really going to protect you from the virus? This one part of it is safety. If they can demonstrate it's safe, they could potentially get away with doing ring vaccinations like we've done for Ebola in Africa, where they just vaccinate people that are in contact with sick people, and they were able to show that worked quite well, also with a limited amount of vaccine, which should probably be targeted towards nurses and doctors, healthcare providers first. But the question, so if it's safe, in a way you can say ethically, you can just give the vaccine, whoever wants it, who is a healthcare provider should get it. But if it really protects them, that will take time to show, because you need to run a clinical trial where you follow people over time, sometimes significant amounts of time, and you try to figure out if they get less of the disease than a control group. So that's what's going on with vaccine. A lot of activity, a lot of companies are in this game already trying to make a vaccine for coronavirus, and I hope something works, at least for the populations who are most exposed to sick people. And the third pillar of the response is therapeutic efforts, and we're working on that one too. If anyone wants to pitch something, it's possible to do that. We have already collected several projects from the CEND associated faculty, and we're working with the Drug Discovery Center on the small molecule approaches, because we have some tools to do so. One of the first projects that we're planning is to actually run all approved compounds in an infection assay with COVID-19, so we can figure out if there's any molecule that can be repurposed. Because if you have a drug that's already approved and the safety has been extensively studied, and it works against the virus, then you could just prescribe it off-label tomorrow. But then you need to generate data to make sure people see that that's actually the case, and hopefully something works. I mean, there's some antivirals in the library. So we're planning to do that. We have already clinical trial for remdesivir, the Gilead drug, is in phase 3. They had developed this originally for Ebola. It's a nucleotide analog that jams up the replication machinery, and they're trying to work on this for COVID. And there's some evidence that it works in some people. I don't know how strong the data is. In the end, they're hoping to release the information April 20th, the results of this clinical trial. So that's going to be very interesting to follow. And some other therapeutics are also being considered. There's quite a lot of work being done on outcome studies, which is very challenging to do if you think about hospitals being overloaded. And they try to figure out what people are taking in the background anyway, you know, patients who just are on asthmatic medication or something like that, and see how they fare relative to other groups. And there was some evidence that, some asthma drugs, basically muting your immune response in the lung was correlating with good outcomes. So that makes sense in a way, because we know that COVID destroys the lung tissue by inducing a really strong immune response in the lung. Even if you've cleared most of the virus, your own body basically destroys your alveoli, and then you lose lung function, and this is what you die from. So it's a valid approach to look at the host response particularly, and I'm proud to have Jeff Cox on the line today. He's a specialist on host-directed therapies and works as a faculty director of CEND also. So we have some interesting things here that could be brought to the table. And other antivirals are used. Chloroquine, you know, we had a lot of debate about it. First tiny study showed some effect with 10 people. Now, yesterday a study came out with 30 people that showed there's no effect. My take on this is like they're both tiny studies. I don't know. I think we have to do a bigger, a better experiment to really say conclusively what's going on. But people are using a lot of things kind of on a hunch that it might work at this moment, and that's, in the absence of better data that we can provide, that's all we can do. And using clinical acumen of the clinicians treating people who may be seeing like certain thing correlates with a better outcome, and then we can investigate in a clinical trial. But why are we doing this today, and why have we chosen the focus areas? The one is diagnostics, because it's critical. We know from modeling studies that with a good diagnostic you can make as much difference to pandemic spreading than with an actual working vaccine. So it can be very efficient if it's used the right way and if it's available fast on a large scale. And thing we wanted to bring to people's attention is also Candida auris. I'll be speaking about this later. This is a it's a hospital-acquired infection, a fungal pathogen that we also don't have a diagnostic for, and we have no drug for, and it's horrible in immunocompromised patients. And the faculty, particularly Anders Nahr, who's unfortunately sick himself today, can't attend, but he's working on that, and he's really worried that this is spreading in US hospitals and worldwide, really, without us knowing, and it's particularly enriching in ventilators. So having some way of testing for that will be really beneficial for the patient community in general, and particularly for the COVID intensive care patients who get hooked up to ventilators. And so the third part we wanted to talk about is ventilators, because there is a shortage, and as more patients enter the hospital and need to be put in intensive care, some go into induced coma, they need to be ventilated, and there is a shortage, like similar to what we see with face masks and other things. But these machines are a bit more complicated, so we wanted to talk about how can we potentially develop a blueprint or ideas for a field ventilator that is kind of low tech, but still does the job, at least in the patients that are maybe not completely unable to breathe on their own, but still can try, to breathe and then get better oxygenated air into their airways. So that's about what's going on right now. I would say globally, everybody is scrambling to get this under control. We have also a project working with Africa with our faculty, Sarah Stanley, taking the lead on that, trying to build diagnostic capacity there. We haven't really talked about low and middle income countries yet, but it is a big need. I mean, Matt is on the call here. He was with us in Uganda many times doing bioscience capacity building, and we've just spoken to them yesterday. They have 120 tests available currently in Uganda for a population of 40 million people. And just today, basically all airlines shut down traveling into Uganda, so it's very difficult to get anything shipped. So we're working with them to try to find some ways to get reagents and testing reagents down there, because they actually quite well set up with machinery. It's just the reagents that are lacking. So this is also where anything diagnostic, there's countries that have very few cases right now and are just at the beginning of this curve, but that we can still make a difference in those for sure, if we could develop ways to test more people and allow for a better quarantine system to be put in place. And hospital overwhelming, in low and middle income countries, situation is even more dire, because even on a good day, there's not many hospitals. And if you need medical care, you need to wait for a long time and travel long distances. So I think particularly there, also the impact of some low-tech solution to ventilators, face masks, things like that would be really impactful. Okay, this is my little overview. I hope to stimulate some thought and discussion. And with that, I hand it over again to Isabelle.